Hello, my name is Jim Rader. It's January 15th today, and I'm preparing for a class we're going to have on January 18th, which is the purpose of this recording, one of many purposes. In this class, we're going to continue from where we left off last time. The class from last time is also recorded and out on YouTube, so you can watch it. This one will be there with it soon. And basically what we're going to do here is we're going to talk about the Y-DNA from a STRS point of view, the earlier point of view. But before I get into that too far, you can see here, this is the front and back cover, the total cover of a book I published last summer. This book captures the complete process that we're talking about over the 4 videos. The problem with the book, it is too brief, it is factual, it's a study. It doesn't explain things enough. And so these 4 lectures will become part of a new book with much editing, and they will try to fill in that gap so that the reader that comes to this material can appreciate it better and understand it better. This book is available on Amazon and other bookstores for about $20. It's also available on Kindle books for $4. If you look over my left shoulder, you'll see several books on the table. Those books, like this book, were published for one purpose or 2 purposes. One was to help me summarize what I'd learned so far and make it possible so that I could come back to this work and pick it up where I left off, read my own book, so to speak. The other one is to get my genealogy off-site so that when I die, the kids won't come in and say, look at all this crap Grandpa has, somebody order a dumpster. Because what I do is I make like 10 copies, and I go to each one of my descendants and give them a copy and say, could you keep this for off-site storage should my house burn down? And by the way, you can read it if you'd like to. I would encourage all of you to do that. In this class, the second of 4 classes offered by the Plumas Genealogy Society, we will cover the next step. The last class, we helped you get comfortable with what the whole process is. What is it we're doing? Why are we doing it? That video, like I said a moment ago, is out on YouTube. If you'd like to watch that full hour class, it's right there. In previous tests, we were testing only paternal line. That's before Ancestry came along with their everything test. In those tests, we started out in the beginning with 12 markers, thinking that would be adequate, and we kept bumping it up to where we got to 111 markers. We're going to look at the tools which help us understand what those tests told us. In this workshop, we're actually going to use actual results. My tests and yours, if you bring them to class, will also add that work to our class discussion. The emphasis on this class has to be on the problem that the majority of genealogists have. The genealogist will tell me, I do not have a Y chromosome, I'm a female. Or they will tell me, I don't have the DNA, the Y chromosome for the person I want to study. This chart shows my great-grandparents. Obviously, there are 2 people in each square on the outside. And you'll see that the Hugheses and the Duffs and the McGees and Stevensons and McIlvaine and Valentine and the Favers, as well as the Ridgers. Now, I am a male Ridger, so I do possess the Y chromosome that came from me, which came from my father, which came from his father, which came from his father. But I do not have any of the other 15 great-grandparents' Y-DNA. Obviously, the females don't have one, but their fathers did. And how do I get these DNA values so I can figure out my genealogy, confirm my genealogy, prove whether I have mistakes in my genealogy or not? That's going to be a large part of our conversation as we move forward through this class. All direct line descendants of a male, like I just showed on the previous slide, have the same Y chromosome. It passes from father to son. If you look at this chart on the left-hand side, it starts at the year 1660 up at the top, and it goes to the year 1960 at the bottom. And those would be the approximate birth dates of these people, and each one of them being a male has received their Y DNA from their father. So the guy whose Y DNA was in existence in 1660 has passed relatively unchanged all the way down to 1960. Now what happens if the guy from 1930 has another child, another son? That son likewise has the same Y chromosome. What happens if his father, the one born in 1900, has a series of sons? Those sons all have the same Y chromosome. And likewise, as we move back up to 1870 and beyond, all male to male to male to male descendants have the same Y chromosome. So we can test any one of those people born in 1960 across the bottom and get the value of the guy who was alive in 1660, because he passed it on. This is a chart, partial descendant chart, showing the descendants of a person named Caspar Raeder, who was born in 1732. In the upper left-hand corner, you'll see that a William Raeder dotted out. He had a son, Samuel, who did not have any sons. But William's brother had grandchildren and others who've passed that Y chromosome down to on this bottom line. You can see Glenn Raeder and Earl Raeder and Roy Raeder and Howard Raeder and Forrest Raeder and others. All of them are candidates for testing for the DNA of Caspar Raeder. They received it from their father. By the way, if you're curious, the program that does this charting is called Charting Companion by Progeny. And if you want to understand more about this, if you go to the company itself, familytreedna.com, their website, you can find this video, which is done by the founder of the company, describing what it is he's trying to provide you, what service.
If you're curious about your genealogy and your anthropology, and you have exhausted the paper trail road, genetic genealogy is the next tool in the toolkit of the prepared genealogist. The reason for this is that you have your history written into your cells. And only now, in 2008, are we starting to be able to read or to decode that mystery which is in all of us. And if you want to know if your ancestors were from Scandinavia before they came to England, because all you know is that your ancestors are English by oral tradition, you can take a DNA test, and if we find that you're in a particular branch of the tree of mankind that is found clustering in Scandinavia, then what we might have been able to do is to push back your genealogy, or at least your story, another 500 or 1,000 years. For most of our clients, that's very, very exciting and totally unexpected.
So that gives you a feel for what the test is trying to do. There are many, many more videos and much more to share on that topic, but that surface view will help you get started. Here's another one. Maurice Gleason, who's a psychiatrist from Ireland, has been doing the advanced Y-DNA tests and everything else in between for some time, and lecturing and leading a group of people in Ireland who've been doing magnificent work. If you watch any of their videos which are on YouTube, each one will bring you forward in your knowledge base. So let's repeat. Males have a Y chromosome. Their father gave them. So every surname you're researching has a male. That's where the surname hangs around, is that male. And he has a Y chromosome that passed down to him through generations. We're trying to find that Y chromosome, figure out what it is, and figure out how he's related to other people who may be cousins. And if you look here at this chart, this is another one by 3D that it's got at the top in small print up here. You've got one man and one woman, the woman in pink, who had many children. And we're focusing on this Reuben Rader. And Reuben married Martha Frances Rader, and he had 3 children. The male over here, a female, and another male. Now, does George Metcalf, this guy down here in blue, he's a male, so he has a Y chromosome. Does he have Reuben Rader's Y chromosome? You can look at Reuben's here. He has a son over here who had 2 sons. That's a male to male to male. So these 2 sons do have Reuben's Y chromosome. But to answer to my question, here, this is a daughter. Reuben didn't pass his Y chromosome to the daughter. If he had she would be a male. So these boys down here have Kenneth, their father's Y chromosome, not the Rader chromosome. And again, here's William and Barbara. They had this whole passel full of kids, the blue all being boys, and you'll notice that all of those boys will have William Rader's Y chromosome. Okay, the concepts in the category of genetics and genealogy. What's helping us figure this all out? What are we trying to accomplish? Now again, back to my example, my Caspar Rader. He was born in 1732, and I have no idea where he came from. I have no idea who his ancestors were. It's like he was adopted. And the information we have is what you see on the screen. But is he really a Rader? Y-DNA 10 years ago with 37 markers confirmed that these 3 sons, who all believe Caspar was their father, have the same Y-DNA. They don't prove that Caspar was the father. There could have been a mailman in the neighborhood that fathered these 3 sons. But we do know that the 3 kids' descendants have the same Y chromosome. So what are we measuring to figure this out with these 37 stir markers? So what do we measure? We're trying to measure that DNA where those mistakes happened so we can figure out the differences between people. There are 2 things we can measure. One is called a STR, S-T-R, and that's an abbreviation for short tandem repeat. The other one is called a SNP, S-N-P, and that's a single nucleotide polymorphism. So the question is, how do we measure these things called STRs? If you look at this cute little curlicue, it has all kinds of information. Upper right-hand corner, it gives you the names of the 4 chemicals involved that we're measuring. The upper left-hand corner talks about how much there is. 559 million bases, 397 genes, 5,000 SNPs. And in the box to the lower right, you can see it says G equals C, not equals, A joins to T. Those are the normal combinations. So if you go down to the bottom left-hand corner of this ladder going across the bottom of the screen, you'll see a letter A in green, and below that you'll see a T. So the A joins to the T. The next position is another A and T, but they're switched, then a G and C. As you go on across, you'll see there's more and more of the same sort of pattern. But there's 2 types of Y-DNA markers, as we talked about before. We have STRs, which are short tandem repeats, and we have SNPs, which are single nucleotide polymorphisms. And this has 2 examples. The STR one with the orange below it is showing TAC, TAC being a repeated pattern on the lower rung. So that would be 3 repeats of the same sets of letters. And the SNP, the SNPs, we will talk about in our next meeting. which will be in a couple of weeks. STR repeats. What do we mean by a repeat? You see there are boxes below. AGAT is in each box. The top row has 3 of them. The bottom row has 6. In between has 4 and 5. Any number can occur at any location. So what we're doing is going through with the DNA test and measuring how many times a particular set of chemicals repeat. How tall is it, in other words? Short tandem repeats is what we're measuring. It's a short sequence of bases repeated many times. Here's a report that shows you some of the STR matching, the short tandem repeats. It has 4 rows down below, which are 4 different men's test results. Up above it has sideways the name of the location. Some have blue background, some have red. The red is generally the ones that change faster. In the boxes below, you'll see on the left column, there's 14 3 times with blue backgrounds, and one person has a 15. So that one person does not match the other 3 on that location. As you go across, you'll see many of them that are clear with no background. That means they have the same value in all 4 locations, all 4 tests. And the less misses you have, the closer you're related. If you take your computer and go on the internet to ftdna.com and log in with the login you got when you got your kit, you will have access to your results. This is the part of the page that shows the things you can get from the Y-DNA test. You'll notice there are many things, including matches, print certificates. Down at the bottom right-hand corner is where you get the actual printed copy of the results. You don't get anything in the mail. You have to print it yourself. And for example, here's my certificate that shows the values that were returned for the 111 marker test. And you'll see it looks crowded. It looks like there's a lot of stuff there, but of course there's over 110 being 111 results. Also, there are other reports you can print out. There's a certificate that tells you about your leading SNPs, which haplogroup you belong in, which we'll look at next time we meet. And if you clicked on the DNA matches, in this case, since I've taken all the way out to 111, I can put the number 111 in that markers box below the yellow arrow. And in this case, what you see are the 3 people that match me on 111 markers. You see the first one matches exactly. The next 2 miss on 1 marker. If we drop that down to the next level test, which is 67 markers, we see the same 3 people. In this case, we have a 0 genetic distance, and we see a person named Andrew James Rader who misses by 1, and another person, Mr. Christopher L. Cook, that misses by 7. And you notice he did 111 also, you can see on his label there, but yet he didn't show on the previous report because he did not match close enough. We go to 37, which is the one we've considered the standard in my study for raters. If you match on 37 exactly, you're related very closely. If you look in this case, I've got 1, 2, 3, 4, 5, 6 people that match exactly. And then I've got 2 that match 1, and then a bunch that match no distance is 4. Now when we start getting down there, I've not been able to do much with the ones below, but who knows what we'll do in the future. You can also go down to the 12 marker test, and this is the one I recommend you do as a filter. It costs $59. It's a cheap way to get in, and you can see in my case I have 15,000 matches there. So if you don't match at the 12 marker relation, you're definitely not related. So you need to go back and pick another person to put in your sample, or however many miss, because you want to have several people that match here at the 0 genetic distance at 12 markers before you spend any more money on testing them. Let's go on to the how to use this thing, methods used in this category. But how close is close on our 37 marker? How should it be interpreted? If you match on 37 out of 37, that's considered very tightly related. It's so close that they say it's extremely close, and it's not likely to happen very often. If you miss one, you still, if you have the same surname, are probably matching. If you miss another one and you share a common surname, you're still matching. If you go to 34, they share a common surname. They're likely to be in the range of matching, but not near as close in time. 33, now we're getting to probably related. And as we go on further down, 32 is only possibly related, and 31 is not related. So you can see as we just cut off one or two on the 37 marker test, we're talking about here 6, which is quite a ways.